Hello, everybody. Thank you for tuning in. My name is Jeff Hung. I'm CCO at Vigene Biosciences. The title of my presentation is Standardized Viral Vector Scale-Up Production and Analytics. I want to thank you for your time in advance for listening in. For those who don't know Vigene Biosciences, we are a Rockville, Maryland-based private company with 17 years of GMP viral vector track records. All of our 3 locations are in Rockville, Maryland. We have altogether 71,000 square feet new lab and office space, including 31,000 square feet GMP space. We are a private company with one of the fastest growing track records, ranked by Inc. 500. We are on the top list for 2018, 2019, and 2020, 3 years in a row. We are integrated viral vector and plasmid vector production and assay company. The picture on the bottom right is a picture of our facility. On the detailed description of our GMP suites, we have 10 GMP suites currently. 4 of them are for viral vector production. We have 1 suite for adherent cell-based, including cell stacks and iCellus. 3 of them are for suspension bioreactor-based production. 1 MCB non-infectious suite, and 5 GMP suites for GMP plasmids. Our DMF will be established by the end of actually September. And we have near future expansion plan to have 2,000 bioreactors capacity in 2021. And we also have limited 2021 GMP slots available. We're going to the technical aspect of this presentation. As a pure play viral vector and plasmid vector company, and we have experience with the majority of the vectors on this slide. And if you look at this viral vector overview, different viral vector have different pros and cons. The manufacturing of a different viral vector are also quite different. So in Vigene, we strive to standardize the viral vector production. And we have 4 proprietary GMP released high productivity producer cell lines, including 2 suspension cell lines, HEC293 and 293T, and the 2 adherent cell line 293 and 293T. We also have 4 patents, 2 issued and 2 pending for AV packaging technologies. We also have proven AV, LVV, and plasmid production processes. We also have ready-to-use GMP plasmids. And as I alluded to earlier, we strive to standardize the manufacturing process for plasmid and the viral vectors. So this flowchart represents the flowchart for a viral vector development and manufacturing. From research grade proof of concept to PD, and if it involves the plasmid, have plasmid production, engineering run, GMP run, and then the final QC release. And in different operation steps, and we strive to standardize the production and including the analytics and the production. And pretty quickly on AAV, there's quite a few platforms one can use for producing AAV virus, including helper virus assisted AAV production, baculovirus AAV production, or helper-free plasmid triple transfection production. And different production strategies have a different pros and cons as listed on the right. And while we standardize AAV production and testing, there are quite a few things we are trying to solve. And so this slide summarizes the overall workflow for AV production and the standardized module we're trying to build. The first one is a plasmid and helpervirus production. The second step is upstream production, including bioreactors and other perfusion fed batch modules, and downstream purification, including both the affinity column and other resins and also columns. The fourth phase is analytical testing. So I'm going to each step of this roadmap and give some description on how we currently doing to standardize the production. So the first one is a plasmid production. And so in Vigene, we have a single-use system for plasmid production, and we use design for quality to make sure the final process and products are fully GMP compliant. So this plasmid production process depicts 5 steps we took. The first one is MCB, testing and the establishment, so that including cell line, MCB development, and the testing and the release. And second step is fermentation, including the upstream production, optimal growth and optimal productivity analysis and development, and then purification. So that one including cell lysis, TFF, and the ion exchange. And then the fourth one is a QC release analysis, including setting up the specifications that meet the CQA requirements, and the final release to perform those tests and provide certificate analysis and the certificate testing. And the entire process is animal component free. So next one, on the second step of this entire production process of viral vector production is upstream. So on this slide, I depict 2 platforms. One is adherent cell-based platform. The other one is a suspension cell-based platform. And both Vigene have experience and expertise and the capacity. For adherent cell-based, we have flatware-based. On this one, we depict the 10-layer cell stack and the iCELLis Nano and the iCELLis 500 for scale-up process. Suspension platforms are represented on the lower row. And if you look at this from the small scale wave to stir bioreactor tank, XDR 10, 50, 200, and XDR 500. And Vigene has accumulated a lot of experience with all of those platforms. For downstream scale-up platforms, we have unit of operation from clarification, TFF, UFDF, chromatography. And it's worth noting that we have developed a fully commercial ready AAV production that also have chromatography-based empty capsid and full capsid separation step. So entire process is fully scalable for commercial production. Analytical development and service platform also is becoming standardized. Molecular, on this slide, I depict 4 major areas for assay development and QC. One is the molecular biology, assays and assay development, qPCR, ddPCR, ELISA, Western. And we also have HPLC, assay development and QC capability, including residual viral particles and other HPLC based, for instance, supercoil plasmid assays. And spectrometry 260, 280, and total DNA is also on here. And the last one, for microbiology-based assays, including the compendial assays and the other microbiology assays. And overall, we have 20 different assays that have been GMP qualified. And so last one, I'm going to talk about this reference standards for AAV analysis. In January 2020, FDA issued CMC guidance for gene therapy. And 3 types of reference standards are recommended by the agency. First one is a certified reference standards, such as USP compendial standards. Second one is a commercially supplied reference standards. The third one is customer synthesized standards. So for the rest of my presentation, I'm going to outline an exciting product line that serves the gene therapy field, that is AAV reference standards. So AAV reference standards that Vigene just launched in September 2020 is to meet the gene therapy field needs to have basically the reference standards for quantification and empty full analysis. The production process for the AAV reference standard is listed on this slide on the top row. Essentially, we use HEC293 cells and using triple transfection, and then purified the empty or full capsids using aldexonol followed by the ion exchange. And then we go through thorough and complete QC release test to analyze those AAV reference standards. And on this slide, I listed all 12 different batches of different kinds of AAV reference standards that Vigene currently supplies, including full capsids of AAV1, AAV2, AAV5, AAV6, AAV8, AAV9. And likewise for empty capsids of AAV1, 2, 5, 6, 8, 9. For full capsids, the payload is CMV, promoter-driven GFP. So this slide just captured the full capsids percentage of each product. For basically the full capsids, the range is from 56% all the way to 82%. And then for empty capsids, the range for empty percentage is from 84% all the way to 99.5%. And this 12 reference standards have been gone through pretty thorough QC release test. This slide depicts the transmission electronic microscopy analyzed the full empty ratio. And this is a TM picture of a representative full capsid on the left and the representative empty capsids on the right. And for the TM, as you may know, the full capsids are white in negative stain. And then the empty capsids are basically have a circle with a dark spot in the center. That's the empty capsids. And so the AV9 is more than 80% full capsids on the left, and for empty is pretty much more than 95% empty. And for this group of reference materials have gone through thorough QC and rigorous QC analysis. This is a certificate of analysis we provide, and including basically the physical particle by ELISA or by, that's for empty, and physical particle concentration by qPCR for full capsids. And then the percentage of full capsids or empty are analyzed by transmission electronic microscopy, as outlined in the previous slide. Bioburden, endotoxin, mycoplasma, and purity are all analyzed. And lastly, I'm going to finish with lentivirus production and to depict the standardization of lentivirus production in Vigene. This is a peer review paper outlining the 4 different companies' production process for lentivirus. So by and large, it's ion exchange chromatography followed by dye filtration. And so we in Vigene, we also standardize the lentivirus production and have a high titer suspension cell-based lentivirus production platform. This picture just shows the high infectivity produced by this production platform. So this is the final slide. I'm just going to summarize our capability and what I described. We have a proven nanovirus AAV, lentivirus, retrovirus, and Plasmid process. We have proprietary cell lines for virus production. And we successfully released more than 40 different GMP batches. And we're a one-stop shop with integrated plasmid and viral vector production. And we also, in this presentation, I also outlined the exciting launch of AV-Reference standards for the gene therapy field. And we have rapid turnaround and short backlog. We have both iCELLis and the suspension bioreactors at our disposal. And we're fully FDA and EMA QP compliant. With that, I'm going to stop and thank you very much again for your time and for your attention.